Now we're going to talk about RNA. If you think about RNA, you're thinking we've got 3 letters here. We've been talking about DNA. RNA looks similar to DNA. So DNA stood for deoxyribonucleic acid. RNA is ribonucleic acid. You start out with DNA. Remember, we talked about the arrows. Arrows is not equals, arrows is yields. So DNA yields RNA or makes, goes to, and then RNA yields, makes, goes to proteins. While DNA and RNA are very similar, they are also very different and have different roles. You need to know how RNA is different from DNA. So there's 3 ways how they are different. First, DNA is double-stranded, RNA is single-stranded. RNA has ribose sugars. If you remember us talking about DNA, DNA has deoxyribose sugars in its backbone, in the brace of the ladder. So RNA has ribose. DNA has deoxyribose. RNA uses uracil instead of thymine. If you remember adenine and thymine, your A and your T went together. Now your A and your U will go together. Uracil is in RNA. So, what are the 3 types of RNA? So, we're talking about proteins, and everything within the cells have to have a protein of some kind to carry out specific tasks. And there's 3 different types of RNA because they all have different responsibilities. Your first one is messenger RNA. It is coded, it is written as M, a lowercase m, and then all caps RNA. It carries copies of your DNA sequence outside the nucleus. So your DNA is inside the ball within the cell, the control center basically inside your cell where everything is kind of going on. It's the brain in there. And so your DNA is stored inside of that. Your messenger RNA is going to carry copies, not the actual DNA, but copies of the DNA sequence. So the order of the DNA, it's kind of like the coding, is going to carry it outside the nucleus. Your R, lowercase, RNA, all cap letters, so R lowercase, RNA, all cap, is ribosomal RNA. It makes up the ribosomes. So you should have now, you've got your messenger that's carrying it outside of the nucleus, and your ribosomal RNA is making up your ribosomes. So you have your transfer. It's the lowercase t cap RNA. It carries amino acids to form protein chains. So amino acids are different types. It's a type of protein. Your amino acids all line up in protein chains, and your amino acids are the building blocks of your proteins. How does transcription protect the cell's DNA? That's what we're talking about. So the transcription is the process of writing it over, and the process of making sure that you get the correct traits, the correct sequence from the DNA, that nothing is altered in the process of changing over and creating new ones. So DNA is essential to a cell's survival because it contains genetic information. So the genetic information is where you get your traits, and your traits provide the characteristics. To protect it, DNA remains trapped in the center of the nucleus except during cell division. So it stays inside the nucleus. All your genetic makeup stays inside that ball of the nucleus in the cell to protect it until the cell starts to divide. So we unwrap the double helix and then we unzip it, and the job of the messenger RNA is to make a copy of the DNA sequence. So remember, copying and pasting, right? So we're going to take this copy right over, like you look at the top and you see all the different, we start over here and we see green, is that blue, green, blue, red, blue, green, blue, red, yellow. See all those different colors? So, and then you see your letters that are inside, right? So it's going to make a copy of this so the message can leave the nucleus without endangering the original genetic blueprint. So that means when you take that copy that's lined up across that's let me see if I can show you, that's lined up right across here. See, you're making a copy right here. You don't want that when you transfer that over to have different colors, to have different sizes, different shades, different letters, because then what you are creating is something completely opposite than what you are intending to create with your new specimen. The process of making messenger RNA copy from DNA template is called transcription. So when you make that copy and you're preparing to make a new sequence, or not a new sequence, to maintain your same sequence in a new DNA, is called transcription. What actually happens during transcription? Well, you have an enzyme that's called an RNA polymerase. It unwinds a small section of the DNA. So the unwinding is the unraveling, is taking it from the twist into something straight, and then it uses one side as a template to make the messenger RNA copy. So whichever that, remember we said that was like the parent gene, right? The parent side. So it takes a copy of that, and then by the RNA copies specific genes by finding promoter sequences at the beginning of each gene. So the promoter sequence is if you have let me see if I can see this. Right here where you have blue, green, green. I can't even see what that says. Is that C? I can't see. Let's just go with the colors. If you have blue, green, green, so then you can tell by looking down here that your blue matches up with your yellow, your green matches up with your red, green matches up. All right, so if you have blue here, what color is it going to match up with? It's going to match up with a yellow. If you have green up here, it's going to match up with a red. If you have green up here, it's going to match up with a red. So the promoter would be the blue, the green, and the green. It's what is initiating the idea of what is needed next to pair up. So your guanine is matching up with your cytosine, so your G with your C. So the G would be if it was the guanine that you were using, that is your promoter sequence. The G, let's say it like this. If you have 1, 2, 3, right? So if you have a 1, you have a 2, you have a 3, that is your promoter sequence. Let's say you have 2, 4, 6. Then 2, 4, 6 would be your promoter sequence. Let's say you have 0, 2, 4. 0, 2, 4 would be your promoter sequence. So in this case of looking for your template, if you have GGT, then GGT would be your promoter sequence. So it's whatever your initial pattern of what needs to be replicated or matched up would be. So the template for making messenger RNA is easy to follow because of base pairing rules. Your A goes with your T, your G goes with your C. And remember, A is adenine, T is thymine, G is guanine, C is cytosine. Whenever the DNA sequence contains an A, however, now you're going to use a U instead of a T when we are talking about RNA. So with your messenger RNA, your A is matching up with your U instead of matching up with the T. If we were talking about DNA, your adenine would match up with your thymine, so your A would match up with your T. But now we're talking about RNA, so your adenine is going to match up with your U, your uracil. This is how it's all going to come together. It's like laying, or let's say building Legos. It's which Legos you are going to put together to match up to make the building that you need to make, how it's going to pair up. So if the DNA sequence is ATT, we have ATT right there. And then we have GAC, AGT, and then CGA. So your promoter sequence would be your ATT, would be your GAC, would be your AGT, and then your CGA. So that's just saying your promoter sequence in threes. The copied mRNA strand would be what pairs up with these. So you would have UAA, because that matches up with the ATT. And then you would have CUG, because that matches up with your GAC. And then I'm going to have to figure this out because I can't see it all on my end. So you have UCA. So UCA goes right there. And then I can't see because it's behind my head. But if you have C, G, A, then your C is going to go with what? A G. And then your G is going to go with what? G is going to go with C. And then your A, because it's RNA, will match up with what letter? Hope you didn't say T. It's U, right? Okay, so that would be G, C, U. Now, what portion of the mRNA is used? So your messenger ribonucleic acid. Not all of the newly created mRNA is required to code for a protein. So we're trying to make proteins. So it's your chains of amino acids that come together, right? Some of the mRNA is spliced out, so it's not deleted, it's just removed from the sequence because it is called nonsense, or it's basically not necessary for that gene. So these unnecessary sections of messenger RNA, which are nonsense, they are called introns, and they are removed prior to the next step in the process, so before you start putting it together. The exons are the remaining portions of the mRNA, which will be used to make the protein. That is a little tricky in my head, so maybe in yours, but this will help you remember. To me, if you're thinking exons, then the X part is what is coming out and what's being removed. In this situation, your exons are actually what is staying, and your introns are actually what's being removed. That's a lot of information to remember at one time, but you need to know this. Make sure you know the terms. Make sure you understand. RNA differs from DNA in its type of sugar, so that's your different sugars from the RNA, your different sugars from the DNA. And then the number of strands, so your 1 versus 2. And then you use your uracil instead of your thymine, so your U instead of your T to pair up with your adenine, which is your A. And the whole process of pairing it up and making the copies and pairing up is called transcription. The introns are removed. The extrons actually stay. Make sure you understand this. If you need to watch it over and over and over to understand it, do so. If you have questions, make sure you ask me. It's important that you understand all of this thoroughly. We will build on what you have learned.